And here's the 9:30, and the clock on the computer says 9:31. So excuse if there's a sound in like a minute or so. I'm Nick Levis. I'm a grad student at the University of North Carolina working with David Fennick. And a central challenge in biology is to explain where the distinctive features that characterize living things come from. Today I'm going to explore one way in which phenotypic plasticity, or the ability of organisms to alter their phenotype to adapt to changes in the environment, might play a crucial role in the origins of novelty. So plasticity-led evolution occurs when a change in the environment uncovers developmental and phenotypic variation that is subsequently refined by selection into a novel adaptive trait. So phenotypes change following a change in the environment. To illustrate this, we'll walk through a hypothetical example. So here we have a genetically diverse population. These different colors just represent different genotypes. And this population encounters a new environment. So it could be a new predator, a new food source. And these different genotypes respond to this environment in different ways. So you see the blue genotype becoming bulgy and round, the yellow one not changing at all. However, now that there's phenotypic variation, there's the opportunity for selection to act on that variation and remove phenotypes and the corresponding genotypes that are poorly suited to this new environment. So here we only have the bulgy blue morph being retained. Over generations, if this phenotype is recurrently expressed and the environment persists, we'd expect the favored genotype to spread through the population and undergo refinement. So here we see they're all blue, and now they're larger, indicating they've gotten better suited to this new environment. Importantly, we expect this refinement to occur in a frequency-dependent manner. So the more often this phenotype is induced and expressed, the greater refinement it should undergo. And this could eventually lead to the evolution of a novel polyphenism, where we have two or more distinct alternative phenotypes associated with distinct alternative environments. It could even lead to the genetic assimilation of the novel phenotype, such that it's expressed even if the environment were to revert back to the ancestral state. So here we have the bulgy morph on the white background, even though that's the ancestral environment. So key aspects of this process include the existence of ancestral plasticity, adaptive refinement of this plasticity in a focal trait, and then potential canalization. And so the rest of my talk, I'm going to walk through a potential empirical example of this process. And we're going to be looking at spadefoot toad tadpoles that normally develop as what you see on the left, this omnivore that eats algae and detritus off the bottom of a pond, and it has a long coiled gut to allow it to digest this material. However, if an omnivore consumes fairy shrimp, pictured in the middle, or other tadpoles, it can develop into what we call a carnivore. As you can see, it has large jaw muscles, a much shorter gut, and highly keratinized pointed mouth parts for handling this macroscopic animal prey. This carnivore morph is an evolutionary novelty restricted to a single genus, Spia. So all of these Bs indicate both morphs are present, and then there's one lineage that might be only carnivores that we'll return back to later. All other frogs, with a few exceptions potentially, have omnivorous tadpoles. And so if we're interested in evolutionary origins of this carnivore morph, we're really interested in this node here prior to when the morph arose. And so to gain information about this morph, we can leverage information and patterns and processes from outgroups, because if any pattern or process is shared in an outgroup and in Spia, it's likely their common ancestor did as well. So the outgroup we focus on is Scaphiopus. So this top group here, they're the closest extant outgroup to Spia. They're ecologically similar in many ways, and they only produce omnivore tadpoles. However, under high intensity competition environment, they will facultatively cannibalize conspecifics, as you can see in this picture. So the first thing we wanted to do was see if we expose these guys to shrimp, will they change some of the traits that we see Spia change whenever they're eating animal prey? So we did that experiment, and I'm just going to jump to the punchline here. There was ancestral plasticity in some of these features that we see in Spia. It wasn't as coordinated, and it wasn't as large a magnitude, but it was there. And so now we're going to step through the next events in this sequence. So what we wanted to do next was see if in ecological conditions similar to those in which we think the trait arose, was this ancestral plasticity adaptive? So we reared Scaphiopus in a high-intensity competition environment in the presence of both shrimp and detritus, with the expectation being if plasticity was adaptive, individuals that switched to using more shrimp should have a fitness advantage. And what you're seeing here is growth and development on the y-axis, and then resource use. So we identified this with stable isotope analysis. Individuals that consume more shrimp tended to grow and develop more than other individuals. So there was potentially an advantage to behavioral plasticity, or at least to using shrimp in this kind of environment. We also competed individuals from families that had different levels of trait plasticities and assessed whether or not any of these SIP-SHIP level plasticities conferred an advantage during the same kind of intense competition. And what came out here was that mouthpart plasticity aided growth during competition. And in a separate study, we also found that individuals with greater jaw width plasticity were more efficient at capturing and consuming a standardized amount of shrimp. So this tells us that carnivore plasticity was potentially adaptive in the environment in which it first arose. However, there was also opportunity for it to be refined further into the polyphenism we see in SPIA. And so that's what I'm going to show you here. This is kind of just recapitulating years of study on the system. We have 4 trophic traits. These dentical rows are kind of hard to see here, but they're little keratinized ridges around the mouth that are used for scraping algae and detritus off of substrates. And as you'd expect, there are more of these on a detritus diet than on a shrimp diet. The jaw muscles get larger on a shrimp diet. The mouth parts become more keratinized. Both of those aid in consuming large prey. The gut length gets shorter on a shrimp diet than on a detritus diet because detritus is less nutritious. So this is a coordinated change in a number of traits that allow this species to capture and consume different resources. And it's different than what we saw when we did this with Scaphiopus. Those responses were not as well coordinated or of the same magnitude. And importantly, this allows SPIA to grow equally well on both shrimp and detritus. So this tells us that SPIA exhibits refinement of this carnivore-omnivore polyphenism. However, recall that refinement of a plastic or conditionally expressed trait should occur in a frequency-dependent manner, such that the more often a particular alternative variant is expressed, the greater refinement it should undergo. So to see if the refinement in SPIA is occurring through this pattern, we're going to leverage information from different lineages within SPIA itself. So 2 main species we study occur throughout most of North America, and when they occur by themselves, so these patterns here, they produce relatively intermediate frequencies of both carnivores and omnivores. However, where they co-occur in sympatry, Spia bombifrons produces almost all carnivores, Spia muscicata produces almost all omnivores. So we have populations that differ in their frequency of carnivore expression. So we can test that expectation that the lineages that produce the morph more often have better versions of the morph than other lineages. So we're really focusing in on this C lineage here, these Spia bombifrons that are almost all carnivores in nature. The first thing we did was collect tadpoles from the wild and look at their carnivore traits. We looked at their mouth parts, their jaw muscles, and overall morphology, and compared them from this lineage to a polyphenic, or a lineage that produces both morphs at intermediate frequencies. And what we found is that these sympatric bombifrons were consistently more carnivore-like in these traits. We also discovered a new trait while we were doing this by chance. We found this palate spike descending from the roof of the mouth. This is not present in the outgroup Scaphiopus, and it was also more elaborated in this sympatric bombifrons lineage. So not only are they exaggerating existing plastic traits, they're adding to the repertoire lineage-specific traits and exaggerating them as well. So this tells us that there's definitely refinement and exaggeration of the carnivore morph in this lineage. But we don't necessarily know if they're better carnivores because of these traits. So what we really wanted to do was compete these individuals against individuals from an environment those from the environment that produce both morphs at intermediate frequencies. So we collected adults from allopatry and sympatry, reared their offspring in common garden, competitive environment on shrimp and detritus, and measured their growth. What you see here, we have allopatric individuals in brown, sympatric in blue, and this blue line is significantly steeper than the brown line. And what it's telling us is that these sympatric individuals achieved greater growth on a shrimp diet than their allopatric competitors. That is, they were winning competition. When we categorize individuals as winners or losers on whether or not they grew more than their tank mate, more often on detritus, the winner was from allopatry, and on shrimp, the winner was from sympatry. Again, what we would expect based on their frequency of morph expression. We looked at a number of things that could be driving this divergence. And the main thing we found was that these sympatric individuals are just better hunters for shrimp. They're better at capturing and consuming shrimp. They're not necessarily better at converting it into new biomass, for example. So we fed them a standardized amount of shrimp, and sympatric individuals captured them and ate them much more quickly than allopatric ones. So this really suggested to us that these carnivores not only are more exaggerated, they're actually better at being carnivores. And so the last thing we were interested in is whether or not this population is trending towards fixation. And so we looked at hatchling tadpoles prior to ever being exposed to the carnivore-inducing cues. So they haven't eaten anything, they're in a common garden environment in the lab, and sympatric tadpoles are hatching out more carnivore-like than allopatric ones. We also looked at patterns of diet-dependent plasticity, first by looking at a suite of genes that are associated with carnivore and omnivore expression. We found in allopatry there's a significant signature of diet, such that individuals fed shrimp have a different gene expression profile than individuals fed detritus, but in sympatry that wasn't the case. And finally, looking at morphology, we see that allopatric individuals in brown had larger jaw muscles on shrimp than on detritus, but sympatric individuals in blue had carnivore-like jaws on both diets. So this is kind of the hallmark signature of genetic assimilation. So these data together suggest to us that these populations, if not fixed, are at least trending towards fixation of this carnivore morph. So to summarize everything I've told you, we have evidence of advantageous ancestral plasticity that's undergone adaptive refinement in a frequency-dependent manner and potentially led to the evolution and fixation of a novel trait. So I'd like to thank my collaborators, our funding sources, the NSF, and all of you for your attention, and I'll take the questions.
So, of the two species, one was, is carnivore and sympatry, the other is omnivore. Is there some obvious reason why the one species went in one direction and the other went the other?
So is there a reason during characterization one became the carnivore, one became the omnivore? And trying to recreate that first contact, there's not a super well-known reason. These omnivores just seem to be better carnivores in the first place. I can't give you a good reason more than that. We did a similar experiment looking at the other lineage, the polyphenix, so they're less carnivore producing, and we didn't find a difference there, but we have a lot of other reasons for that expectation that I can talk to you later about if you want.